now. So as and when the audience, as and when they come, they will join, and so let us start the talk. Welcome everyone. A very good evening here at IIT Kharagpur in India, but a very good morning there at USA. I believe you're in Cambridge, right, Dr. Chowdhury?
Yes.
Okay, so a very good morning to you and your colleagues over there. Today we are gathered for an exciting talk on development of evolution-informed drugs, Darwinism means therapeutics, by Dr. Sourav Chowdhury, and this is being organized by the IEEE Computer Society Student Branch Chapter here at IIT Kharagpur. My name is Sarkar, and I am the current chair of the IEEE Computer Society Student Branch Chapter, and also I am a PhD research scholar at the SWAN, that is Smart Wireless Applications and Networking Laboratory at IIT Kharagpur. I'm in the Department of Computer Science and Engineering under Professor Sudeep Misra, and I will be your host for this particular talk. And I would like to welcome you all to this wonderful event. Now, before Dr. Chowdhury begins, I would like to introduce him to our audiences. So, Dr. Chowdhury did his PhD in Biophysics and Structural Biology from Indian Institute of Chemical Biology, CSIR, which is under CSIR in Kolkata. After that, he did his he was a senior postdoctoral research associated in evolutionary biophysics from Harvard University, and he was also a senior research associate in the Laboratory of Systems Pharmacology in the Harvard Medical School. He served as a scientific advisor in computational and systems biology at Zion Therapeutics, and a very great news is that he is joining as an incoming faculty at BITS Pilani-Hyderabad campus from May 2024. So, Dr. Chowdhury, I would like to congratulate you heartily for getting this faculty position. And let me also tell you that BITS Pilani-Hyderabad campus is also my alumni. So, I did my master's, integrated BE and ME from BITS Pilani-Hyderabad campus itself. So, it's very heartening to know that you are joining my alumni. I hope to go there someday and meet you in person.
Sure.
So, going back, so Dr. Chowdhury did his research on his areas of research lies mostly in computational and experimental studies at the interface of protein folding evolution problem, evolutionary systems biophysics, and evolution medicine. So these are very exciting terms that I see from coming from the medicine and the evolutionary perspectives. Now, Dr. Chowdhury has also achieved quite a bit in his research and in his research career. So he was awarded as a Young Scientist Award at the MAWA Medical Advances Without Animal Prize. He got the Best Presenters Award at the International Conference on Intrinsically Disordered Protein at Aizer Mohali, held at Aizer Mohali in December 2017. He won the Karlstrom International Diversity Award by Gordon Research Conference Committee, Excellence in Research Award by CSIR for his outstanding research. He is currently in NIH to support. He is currently awarded by NIH to support his postdoctoral work at Harvard University, and he also won the QSP Prize, the Quantitative System Pharmacology Session 23, which was jointly organized by Applied Biomath and MIT. So before I hand over the session to Dr. Chowdhury, few general instructions to the audiences present here. So please keep yourself muted during the talk and keep your cameras off as well. Please wait till the end to ask questions. There will be a separate Q&A session at the end. An attendance sheet will be put through the chat box. Please note your attendance there. If you may type any query or issue that you have in the chat box while the talk is in progress, and we will be taking a group screenshot of the event at the end. So at that time, we request all of you, if possible, to please turn on your camera when we ask you, so that we can take the screenshot. So with this, I would like to hand over the session to Dr. Chowdhury, and so you can start now. You can present your screen and start the talk.
Thank you so much, Konteo, and very nice to get the invitation and be with you all. And regarding the questions, it would be great if because sometimes it's a little hard to see from the chat box, if some of you can kind of essentially recite the question, that would be nice.
Yeah, so for the question, I would like to request the audience to please if you have a question, please note it down in somewhere in notepad or something. At the end, we will have the Q&A session where you can, unmute yourself and you can ask directly to Dr. Chowdhury. So, yeah.
Great. Okay. So you can see my screen, right?
Yes. And I hope this is being currently streamed live as well. Susanna, you may just type in the chat box that with an affirmative tone.
Great. Okay. Thank you all for all these logistics at the back end. Yeah, great. And yes, so very good morning. I mean, it's essentially not a very bright morning here. It's a lot of a gloomy morning here. And good evening to you all back in India. And thanks for your time. So today we will essentially, in the next 35-40 minutes of discussion, it may slightly go beyond that, but I'll try to not rest through the slides. I'll try to be slow. I hope to be within the time frame. Okay, so in today's discussion, it would be, given we have an interdisciplinary audience, I'll try to make it sort of less jargonish and essentially, as I mentioned, so that please stop me whenever you feel like, so that we can have conceptual clarities when it comes to certain terminologies. And this would be sort of kind of at the interface of evolutionary biology, biological physics, and some of the tools which we kind of routinely use, not just the tools, but also the ways to have an abstraction of the problem from statistical mechanics and computer science. So, but primarily it's going to be a biological problem, and the question is pretty biological and clinically relevant. And as from the title, it is kind of pretty well evident. So essentially, we are trying to utilize some of the concepts coming from Darwinian biology, precisely more of the evolutionary biology, to sort of inform our drug discovery frameworks. And we will essentially understand, why evolution is kind of an important, why looking through the prism of evolution essentially helps us to understand the problems associated with drug discovery and some of the recent challenges with many of the disease biology problems, and how essentially evolution-informed strategies can help us to have better understanding and sort of a more promising solution. So yeah, so currently I work, I'm with Eugene Shaknovich here at the Department of Chemistry and Chemical Biology. So I'm primarily associated with in Harvard we have two campuses. I'm primarily associated with the Cambridge campus, which is more of the basic science campus. And I also have a second affiliation with the medical school. Essentially, I'm a part of a consortium. It's the Harvard Program in Therapeutic Sciences, and one of the sub-consortium is associated with all, the applications of systems biology tools to sort of address some pretty pressing biological and clinical question. And that's the consortium we call as the Lab of Systems Pharmacology. So yeah, so let's get started. So we will essentially, before we come to this concept or this premise of evolution-informed drug, we'll essentially try to understand what led me and also what led sort of the protein folding community to essentially get excited about this this evolution-informed strategies. It's also kind of my own personal journey. So essentially, it's all about understanding. Initially, it all got started with got started to understand, essentially through a process to understand how protein molecules fold. Proteins, to make it a little more clear, these are kind of gene products, and proteins are kind of molecular machines, and they're responsible for all the majority of the critical biological processes inside cells. And they need to adopt a certain three-dimensional structure to be biologically relevant. And how it happens and through what processes it happens, what are the substructures essentially formed during the process of these kind of final structure attainment. Essentially, everything falls within this domain of this protein folding, and it's essentially a problem. We call it protein folding problem. And there is a clean link between protein folding and protein evolution. And technically, it's what we call foldability, evolvability link. And for me, and also largely for the protein folding community, this has been there has been some precise areas where protein folding problem is kind of having particular relevance, especially, how fast and essentially the sequence in which the structures essentially form. And in that context, neurodegenerative diseases is one step. So we'll talk a bit about that. So we'll try to understand this link between protein folding, protein evolution, which is sort of an inspiration as to how the protein folding community got interested in these kinds of evolution-informed strategies, or like therapeutics, which are evolution-informed. And then we will try to understand this problem of drug resistance, which is where these evolution-centric approaches are kind of extremely promising. And we'll have some numbers. Essentially, this drug resistance problem is indeed an alarming problem. And we will primarily focus on antibiotic resistance problem, because that's one of the model systems on which we have been working. And in principle, it's something that the frameworks which we are developing and the approaches which we are taking can be extended beyond the antibiotic discovery. It has a more generic use, and we in principle can take them to address the problems of drug resistance which we see in evolving cancer population. But we are essentially currently focused on antibiotic resistance problem, and that's one of the model systems on which we are working. So we'll discuss on that. And then we will essentially understand when we say protein evolution and when we say drug resistance is an evolutionary problem, we'll see what evolutionary landscapes literally look like, and how it can essentially provide us with the information which are needed with these kinds of framework discoveries, like the ones which we have been talking, like these evolution-informed drug discovery platforms. And also another important thing, which is not just particularly relevant not just only or in particular relevant to these evolution-informed approaches, it's kind of relevant to drug design and the mechanism of actions for the drug. It's essentially the problem of off-targeting. So many drugs which we consume, sometimes we tend to consume drugs in combination as a part of the prescription medicine. So generally they do have off-targets. They essentially have multi-targeting problems. So essentially drugs are designed to kind of interact with a particular target protein, but maybe inside the cell, what happens is that probably it's kind of interacting with multiple proteins. So essentially the phenotype which we see, which is likely to be, addressing some disease or maybe regulating certain disease phenotype. So that phenotype could be, could be essentially a summation of the effects coming from these multi-targeting. It's not purely one molecule contributing or one target protein contributing to the phenotype. So we very recently, it was an active collaboration with UCSD, with the Paulson group, I'll essentially talk about that. We came up with a framework whereby we can actually demystify, we can understand the intracellular mechanism of action of drugs, and precisely it is kind of helping us to understand the possible off-targets. And that's something which essentially was a spin-out from our original project where we were working on this evolution-informed drugs, but it is now having a more generic use. Essentially, it's a framework which can be used to essentially understand off-targeting and multi-targeting. And then in the end, we will essentially talk about the possibilities and essentially some exciting possible avenues which essentially can be explored using this evolution systems approaches or evolution-informed systems approaches. And it sort of falls within this domain of kind of a very interesting and exciting domain of synthetic biology. So essentially, these are going to be the major components in our discussion. So we'll start off with protein folding, protein evolution problem. We'll go into the resistance. We'll understand the landscape and essentially talk about the evolution-informed strategies, and then finally with the futuristics, essentially synthetic biology and systems of biology approaches. So a little bit about my academic training. Why this is important? Because this also essentially is a way, it's not just my own academic training, but as I mentioned, that how this also kind of explains how or shows how protein folding community in general got interested in this kind of, in this new age therapeutic discoveries, which are primarily protein evolution informed. So I had my PhD at the Indian Institute of Chemical Biology with Dr. Krishna Chattopadhyay. So my PhD area of work was primarily protein folding problem. But the model system, the model protein on which we were working, we also had an evolutionary angle. So essentially, we would discuss on that. So essentially, we were working with a protein system which is associated with neurodegeneration, and certain mutations are particularly associated with the early onset and essentially more aggressive forms of this aggregation, or this neurodegenerative disease. So we wanted to essentially have an evolutionary insight to that. I mean, essentially, why these mutations happen, which among these mutations are evolutionarily more commonly sampled. And then we wanted to have an integrated understanding, like essentially integrating this evolution-centric information, which essentially talks about these kinds of emergent properties, like which mutations generally emerge more rapidly compared to the other mutations. And then we essentially went on to map these mutations on the protein structure and deployed both computational and experimental techniques to essentially have a more meaningful picture. So it was primarily my PhD area of work, and exactly this was something which I got started with Eugene. So here at the Department of Chemistry. So Eugene's work is primarily at the interface of evolutionary biology and statistical mechanics, and our group. So our group was more of a theoretical chemistry group. So we try to formalize this problem and essentially try to come up with a theoretical understanding of these kinds of evolutionary events, and then it helps us to essentially understand, based on the past mutations, essentially the way a protein has evolved and mutations have been sampled. These kinds of, the frameworks which we develop in our group, or historically which we have been developing in our group, in Eugene's group, it kind of helps us to sort of predict what kind of mutations can happen in future. So based on that, it could be, and that information can be essentially integrated to drug discoveries. And I will show what I mean. So Eugene's group, so it is sort of at the interface of statistical mechanics, evolutionary biology, and core protein physics. So this is also a little bit about my own academic training, and also how how I transition from protein folding, protein evolution, and now more into this drugs, or this evolution-informed drugs. So as I mentioned, so the protein with which I was working in PhD was superoxide dismutase. So as I mentioned at the outset, again, to make things a little more clear, proteins are gene products. And this gene product, superoxide dismutase, is one of the critical proteins associated with it's one of the critical proteins in the cellular antioxidant defense mechanism, or antioxidant defense cascade. And its primary function is to essentially get rid of free radicals, to be more precise, superoxide radicals. Now, essentially all oxygen-based life forms, essentially what I mean, like cells which essentially use molecular oxygen as the terminal electron acceptor, they're all primarily prone to these kinds of free radical induced stresses. And that is the reason as to why cells are precisely armed with the antioxidant defense cascade, and superoxide dismutase is actually one of the key molecules. It's actually the first molecule in the entire cascade. So this molecule has also a very interesting evolutionary history. It essentially originated in those forms of cells where oxygen-based metabolism, or oxygen-based metabolism had originated. So it has its origin in the cyanobacterial life forms. Essentially, it happened during a time of biogeologic evolution, when molecular oxygen was essential, when Earth's atmosphere was sort of getting saturated with molecular oxygen, and cells had to use molecular oxygen and had to integrate it with cellular biochemistry. So essentially, it's like a way, this is more like a way in which the toxic effects of molecular oxygen could be essentially detoxified. This antioxidant defense cascade is primarily associated with that, and SOD molecule is actually the key molecule associated with that cascade. So that's more on the functional side of SOD, and this is how the protein actually looks like. This particular protein is a metallated protein, so it has copper and zinc as metal cofactor, and the metals essentially creates a electropositive core, which is actually associated with, it acts more like a sink. So the superoxide radicals, it undergoes different kind. It keeps on colliding with the molecule at the surface, and then just funnels itself towards the electropositive core, which is where we have these metals. So this protein, as we can see here, it's otherwise, and these are the beta sheet regions of the proteins. This is that it has a little bit of alpha helix in the protein structure, but primarily the protein also has a lot of these unstructured regions. This means that these are the regions in the protein which has very low bias towards the structure, so it can adopt different kinds of conformation that way. And it's not a very rigid beta sheet or alpha helix kind of a structure, but the coordination of the metal actually provides it a structure. So what actually happened is this, that the presence and the absence of the metal, and these are essentially the coordination sites for the metals. Essentially it is having histidine residues inside the metal, inside the protein. So this coordination sites, and upon coordination, I mean, when the metals get integrated, the metals get coordinated to the protein gets very stable. The moment you take the metals out, the protein is relatively less stable. And as a result, then that actually allows the protein to adopt different kinds of conformation. And what can happen is this, that when it is doing such things, so it imprints it. So these are precisely what we call as dimer. This is a monomer, this is the monomer, and these are like a homodimer. So what happened under those kinds of conditions, that these proteins can essentially start stacking. And that's what in technical terms we call the problem of protein aggregation. And protein aggregation has been implicated in multiple disease conditions and with many neurodegenerative diseases, including Alzheimer's and Parkinson's and ALS. So the reason we picked up superoxide dismutase, of course, it had a very interesting evolutionary context, and at the same time, it is also, this protein is actually one of the causal factors behind ALS. Now, what is ALS? ALS is amyotrophic lateral sclerosis. We are all familiar with the picture of Stephen Hawking. So Stephen Hawking was actually suffering from ALS, and it is a neurodegenerative disease. It's essentially more of a muscular neurodegeneration, and later it leads to losses in kind of neurocoordination. So there are multiple factors, not just SOD there, but SOD1 is considered to be one of the prime causal factors behind ALS. So we wanted to understand essentially how the protein essentially folds up. So we had relatively a simpler perspective towards that. Why simple? Because there were already known information associated with that. So we were primarily interested in how the molecule folds up, and during the process, essentially what kind of conformations are actually being sampled by the protein. And that's what we essentially say as like understanding the conformational aspect of superoxide dismutase. Why this was important? This was important to us essentially to understand different events associated with metallation. As I mentioned, as I showed at the outset, that based on the fact whether the protein is having metal or is not having a metal, more importantly, it is a bimethylated entity. So whether the protein is having one metal versus two metal, which methylation is happening, it kind of decides, it to a large extent decides what kind of structure the protein is going to have. And that in turn would essentially also dictate whether that particular structure of the protein would function as a seed for these stacking events, or what we technically call as an aggregation event. So it's more about understanding the protein folding problem and folding in the context of superoxide dismutase. Then we were interested in understanding essentially, okay, if there are multiple mutations, then to what extent these mutations can be explained from an evolutionary perspective. Because a protein, when a mutation is happening and a mutation is getting fixed, it means that the protein is not completely losing its function. It is definitely, otherwise, because if a protein is too much destabilizing, then with a mutation is too much destabilizing, then the molecule would not just fold up. So there's something happening, which is why the mutations are actually getting tolerated, which is why the mutations are actually getting sampled. But all these kinds of, these later complexities are kind of emerging. So essentially the stacking and aggregation things are happening. So we wanted to have a better understanding of for these kinds of mutational events. That was primarily through the prism of evolution. And then we integrated them. And like understanding these conformation sampling and also the biases of certain mutations, essentially understanding how one region of the protein is evolutionarily coupled with the other region of the protein. Because essentially what happens because essentially when the protein molecule folds up, it essentially starts off as a linearized polymer chain, and then it keeps on going through the process, which leads to the molecular structural compaction, and as a result of which distant residues sort of come closer, and these are essentially those pairwise interactions. So generally, in the entire process of compaction, how the distant residues are coming closer, it plays a very important role. Now, let's suppose there is a mutation in one of these residues. Essentially, let's say 23 and 55 residues are now pretty close. Now, there is a mutation in the residue 23. So the 55th residue would not actually have its pair with which it was actually interdependent. So the pairwise interaction is now going to get destabilized, and that's going to have an effect on the structure. So we actually went on to map, as I mentioned, the known mutations in the context of SOD, where they're located, and how they're evolutionarily kind of sampled. And then we used our evolutionary algorithms to essentially understand these kinds of positional interdependencies, and that actually helped us to sort of capture evolutionarily constrained region in superoxidase mutas. And finally, as I was mentioning, that we integrated these two, and that we went on to sort of understand that how it kind of controls this switching. I mean, the protein molecule was supposed to fold up and perform its function, but it's not doing that. So there's some subtle, it's more like tuning different knobs here and there. So like some of the knobs being tuned allows the protein still to fold, but some doesn't. So what leads to this switching, and what are these critical episodes or in this entire process, which are kind of, responsible for these kinds of folding aggregation switching events. So these were the works which we eventually published, so these were like series of works which we published. And as we can see over here, this kind of reddish, orangish, kind of reddish area is essentially the region inside the protein molecule, which are kind of under tight evolutionary selection pressure. So this is where we used mutual information-based kind of analysis, which was where we were actually looking for positional dependencies. So essentially, if we have two positions which are highly interdependent, when we go to map them, we set certain cutoffs, like essentially a residue, a position in a polymer chain. If you think of like the immediate next residues, we always would have higher kind of we will always have higher MI scores, but that doesn't mean anything in the context of these kinds of distant residues coming closer. So we do have some positional cutoffs and distance cutoffs, and based on that, we kind of we essentially try to understand where are these kinds of interdependent positions where they are clustered. And what we found pretty interesting, that although this region of the protein, which is otherwise pretty unstructured, but actually has a lot of these interdependent residues. And these are actually the positions, and these are actually those substructures which are also responsible for methylation. And as I mentioned, methylation is not just important for the functionalization of the protein, it is also important towards the structural stability. So we essentially had an evolutionary explanation towards some of this mutation, and then we went on to generate a mechanistic model, and we sort of compared which among the two metals are essentially a kind of more which of the two metals are actually more in a way more decisive in terms of deciding whether the protein would fold or it would kind of aggregate, and that's what we can see. So this is essentially copper. So this protein is more of a cell cytosolic protein, and if we can go back to our life science textbooks. So essentially, this is all we are talking in the context of human neurons, and but in general, because superoxidase mutas is essentially is found in all human cells, so this is more of a cytosolic event. And essentially, in cell cytosol, in emergence, we have the membrane. So essentially, this metal uptake happens with the help of another protein, which we call as chaperone proteins, and these are copper chaperones. So essentially, what happened, like this copper entry is associated with like kind of making the protein kind of semi-stable. This is a copper coordinated state. Now, beyond the copper coordination, what happens, like this is actually mediated by another protein. So that actually provides the internal environment for the second metal loading, which is where this zinc loading happens. And that leads to the maturation and the functionalization of the protein. And in principle, the protein can do its actual function, which is essentially getting rid of detoxifying the cells from the free radicals. And if it doesn't happen, which is where we used all this mutation information, we actually observed this was of course like a combination of both computational and experimental studies. We carried out force microscopy, AFM, it's more of like understanding these aggregation, the structures of the aggregations, and what we actually observed, that this is actually that key event. And if there are mutations which essentially restrict the entry of zinc, then the protein would still be in a copper bound state, which is okay, which is kind of a semi-stable state. But this is actually a particular state when the protein is kind of semi-stable, but at the same time, it actually exposes residues which are more of like partly hydrophobic, and that leads to more of these kind of stacking things. And aggregates start forming. So essentially it starts to function more as a seed. And all these things, as I mentioned, happens as it is happening at the periphery, at the inner periphery of the cell membrane. So these aggregates essentially then start to essentially stack onto the cell membrane, and that leads to pore formation and leads to membrane depolarization, and ultimately that leads to cell death. So this is kind of that critical a critical state, which is partly a semi-methylated, monomethylated state. So we had a mechanistic explanation for these kinds of events. We finally generated this picture, which was kind of a combination of combination from the information we got from the SOD, this molecule's folding landscape, and also how, like, what mutations are kind of critical, and that's how we could essentially zoom in to these metal pockets. It was kind of an evolution-informed strategy to essentially understand the mechanism, because that's what allowed us to essentially zoom in and informed us to zoom into this metal pocket, which is where we have these interdependent pairs, which is primarily coming from this covariation, or technically this mutual information-based analysis. So this was where we were not, this was still like a very mechanism-centric story, and in the context of neurodegeneration, not in the context of resistance. But we found, like, and this is not just us, I mean others also are kind of deploying these kinds of integrated strategies. We are finding that these kinds of evolution-centric ways of looking at these folding aggregation problem provides us with a better understanding, and it in principle helps us to kind of deconvolute these kinds of, if there are 5 episodes essentially, like as we see here, like the methylation event, it kind of informs us to essentially focus on specific events, because we know that they are under evolutionary constraint, and that's what we actually did over here. We could compare the copper uptake versus zinc uptake, and we could actually say, and we could actually inform, and it was pretty well validated by experiments, that which among the two methylations are kind of critical and leads to aggregation, and which ultimately leads to cell death. So this was exactly the kind of what this was back in 2018. Our lab, my PhD lab, still keeps on working on that. We also keep working, but this was a story which I was presenting in Gordon conference, and which was where I met Eugene, and which was where I also heard, there were multiple sessions on these kinds of, taking these evolution-informed approaches to problems which are of more relevance to disease biology. Of course, neurodegeneration is, of course, one such thing. But when we think of problems like drug resistance, which is purely an evolution problem, it's something where you can actually use this kind of strategies. And in principle, it's not just about understanding the mechanism. We can inform the drug discovery protocols that way. I mean, how we should design drugs, how we should think of drugs. So a little bit about the numbers, as I mentioned. So this is one of the critical threats, and another pretty problematic part of the problem of antibiotic resistance in particular. This is particularly a problem with antibiotic resistance, that since it is an evolutionary problem, and no matter whatever the kind of traditional antibiotics or even the new antibiotics which are coming up or one consumes, the community will, this is more of a recurring problem. The community would always keep facing this problem of resistance because it's more of an evolution problem. It's like you're putting stress, and essentially the evolving cell will always find a way to escape that stress. So it's more of an escape strategy. So unless we understand, so evolutionary landscape or analysis of evolutionary landscape precisely helps us to understand the escape mechanisms. And as we take a very protein-centric approach, it actually helps us to, so if we have a target protein, which is what this antibiotic molecules target, and if we know that what are the mutations essentially contributing to this escape strategy, we can come up with an anti-escape strategy, and that's what we will kind of discuss. But the numbers are literally alarming, and it's not, and as one of the this World Health Organization report suggests, it's not just, I mean, in the early 2000s, it was more like a prediction, and people were talking about, okay, this part of the world, that part of the world, especially in those parts of the world where, again, you have, like in the US, you just cannot go to any of we cannot go to these drug stores and get antibiotics. One needs to have prescription. But in other countries, like essentially we can just purchase antibiotics, and we don't need prescription. So this indiscriminate use of antibiotic is one of the causal factors. So people were kind of segregating the problem based on these kinds of factors, more of sociological factors. But increasingly, what we are kind of experiencing, it's more of a global problem. It's very hard. Of course, there are certain countries which are more prone to these kinds of problems, but it's more of a global problem. Another reason as to why it's also a big global problem, that now the biopharma companies are sort of deviating from, their funding strategies are more and more getting deviated from antibiotic discovery. Again, the problem is at the root. Essentially, it's more of an evolutionary problem. And so whatever be your solution, the problem is unless we understand the escape mechanism, it's very hard to come up with some kind of a semi-perpetual or perpetual solution. So that's, and also another thing, that the number of drug targets inside a bacterial cell, the druggable proteins are also limited. So not all proteins are druggable. So there are multiple factors which is kind of bottlenecking this effect, and which is why the biopharma investment is kind of going general, which is where we have this huge problem of evolving cancer cells, which are also kind of getting which beyond a point of time essentially emerges with tolerant phenotype, resistant phenotypes. So these are some of the mechanisms as to how resistance in bacteria sort of happens. It's again kind of for old textbook things. So what happened, like, it's to tell very very generic way. So either the drug fails to enter, so it is more like a decreased permeability thing, or the drug is entering, but the bacterial cells are just pumping out the drug. This is a very general way in which the resistance essentially alter the target. So if we know, so generally the antibiotic molecules target a specific protein, and the molecules are, let's say, it kind of mimics if it's an enzymatic protein, and it's an essential protein. So essentially, what happened, that the compound, essentially, or the antibiotic compound, mimics the structure of the structure of the enzyme's actual substrate, its biological substrate. So what happened? It just competes with the biological substrate and blocks the protein's active site, or essentially the enzyme's active site. So as a result, the enzyme, which is otherwise functionally extremely critical for the cell, is now kind of in its defunct, immobilized, and kind of in a bound state, and it cannot actually do its function. So this is kind of a very common strategy as to how the antibiotics are designed. And the most efficient way by which bacterial cells act, and this is also something not just limited to bacteria, it happens to any evolving cells. So if it's essentially what happens, the target protein gets mutated in a sense, like the antibiotics interact with the protein, and for the interaction you need there needs to be certain chemical interactions, so there needs to be some residues which would participate in hydrogen bonding, some residues participating in van der Waals interaction, some residues just providing a physical flexibility in the pocket. So it's more of a kind of a concerted activity happening in the active site, which involves, let's say, 10 to 12, based on how big the molecule is, the compound is. So 10 to 15-ish, amino acids participating in this entire process. And if there are a mutation, so that would sort of destabilize this interaction, and that would actually allow the protein to essentially evade the drug action. So the molecule would not be able to bind, and the cells can grow. So this is a very common strategy, and we were literally interested in that. Another thing is, like, the molecule can get inactivated. So that's another way in which, like, many of these penicillin family proteins, penicillin family of antibiotics gets inactivated. But the evolutionarily most efficient way and the most specific way to essentially, and given that evolution is more of an optimality solution, there are multiple moving components, but the one which actually allows the cells to have, the best emergent property, which is essentially the evolved phenotype, is essentially to alter this, the target protein. And we were primarily interested in that. So we get into the traditional part. So essentially what happened, as I mentioned, so the drugs are designed against against some specific target proteins, and generally these target proteins are kind of essential protein, because if it's a non-essential protein, inhibition of a non-essential protein would not actually impact cellular viability. Okay, cells can grow a little slowly, but cells would not die. So essentially, that's one of the critical criteria. Traditionally, people used to think about, or even now, when we think of typical drug discovery from from a chemist's perspective, it's like finding out a target protein, and one of the critical criteria for a target protein to be druggable is like to what extent it is essential for the cell. So for that, there are certain pathways, certain metabolic pathways, which are kind of enriched with these kinds of essential proteins, and these are those pathways which are either associated with essentially like the utilization of like the energy sources, essentially the central carbon, but then like the carbohydrate metabolism. These are more of super pathways associated. These are like multi-protein assemblies, and so these are like those kinds of supramolecular associations. Another critical pathway which is also which is kind of extremely which is extremely critical for cellular viability is actually the pathway associated with DNA metabolism. And this is actually this folate pathway, which I'm actually talking about here, is one such pathway which has many proteins which are very essential proteins. If you knock them out, if you inhibit them, then essentially cells would not grow. And it's not just this folate pathway folate pathway is one such pathway. Essentially, it's a part of we can think in terms of network. It's actually one of the subnetwork which is a part of a bigger network of metabolic proteins which are associated with DNA biosynthesis. When a cell has to divide, the DNA number has to get doubled, and then the cells will essentially sort them. So folate pathway is one such, and folate pathway is also targeted even during the cancer therapeutics, or even during the anti-cancer therapeutic regimes. So, but there are certain proteins which are selectively present in the folate pathway. So, if you think about, because this is also important, if you're thinking about a pathway which is an essential pathway with a lot of proteins which are essential proteins, and if that pathway and the proteins exist in humans as well as in bacterial cells. Now think of an infection situation, then we're kind of using a drug, and the drug is kind of now interacting with both the human protein as well as the bacterial protein. But we don't want to have this interaction with the human protein happening. We want to have interaction happening with the bacterial cell. So we need to have a selectivity. So, which is why we look for proteins which are either selectively present. So, architecturally, the pathway could be a little more complex in human, but but functionally, let's say both in humans and bacterial cells, they're doing the same thing. But we may have a particular protein or some subset of proteins which are selectively present in bacterial cells. So that could be a target. That could be a way we can look for drug target. Other thing is this, like we can have the same protein, like the one which we will talk here, but the protein is structurally slightly different. So that also allows us, to have this aspect of selectivity, and we can think of drugs and selectively targeting only the bacterial protein, not the human protein. So again, this is essentially the way that the traditional drug discoveries happen. So folate pathway is one such, and we have a particularly interesting protein over here, this dihydrofolate reductase, which is actually a pretty interesting model system, and also it's an essential protein which has been used as a drug target, and because there are known formulations like trimethoprim is something which we use even in clinics. We use Bactrim as kind of as a combination of trimethoprim with another drug which is also a sulfur drug, and this is kind of one of these commonly used antibacterial formulations. So everything is fine. Trimethoprim is actually a pretty stable inhibitor. I mean, it's a pretty potent inhibitor. It actually inhibits dihydrofolate reductase. But trimethoprim, since it has been used for many years, we have a lot of data associated with trimethoprim. So it's very effective, but at the same time, what happened, like trimethoprim is also notoriously associated with the emergence of these kinds of mutated protein variants. So essentially what happened, like TNP, this trimethoprim compound, it is more of a substrate analog. So it essentially blocks the entry of the substrate, it outcompetes the substrate and inhibits the function of DHFR, dihydrofolate reductase. But DHFR undergoes active site mutation. It's what we call as the escape mutation. So it precisely escapes the action of trimethoprim. So on one hand, it's very effective, but at the same time, it is also notorious with these kinds of, as it elicits these kinds of escape pathways, and as a result, we have these kinds of escape variants coming. So it is not just what we observe in a laboratory strain, it's also what is observed in clinical isolates. So DHFR that way is a very interesting model system because of all this information, and all this information could be in principle mapped, and we can generate an evolution landscape. And all these things can, in principle, inform us about the fact that how DHFR in principle evolves, and we can have an anti-evolution or an anti-escape strategy based on these kinds of information. So this was actually our motivation, and this is what we actually in evolution informed strategies, or as a part of evolution medicine, we always, this is more of a depiction in general for any kinds of when we have this issue of this fitness problem. So the fitness is essentially a kind of a thing which is used. Again, this is a terminology which has different kinds of a visual, but when we think of fitness in the context of statistical mechanics and evolutionary biology, it's actually a way. So there could be multiple ways we can actually compare how fit a particular species is. If we think about a wild-type cell, which is where the antibiotic is functional, and a strain which is kind of emerging, which is essentially evolving with mutations after a certain days of treatment with the compound, and we can compare them using some numbers, and those numbers essentially kind of define the fitness, and that could be like how fast the cell is growing, or there could be other things essentially. In particularly for bacterial strategies, we actually use this parameter, like how fast the cells are growing, and we use that to compute the fitness, and we actually compare the fitness of the wild type strain with the evolved strain. And since we have multiple mutations, so essentially these are all going to be, it's basically a journey from one peak to the other, and that goes through a valley. And so essentially if we have a wild type, it's actually at a peak. And so now based on as the kind of selection pressure we are putting, the mutation is happening and the protein essentially hops from one peak to the other. And since we have multiple such mutations, we can actually compare these fitness, essentially the peak heights, and we can have idea about the fitness. And why this is important? Because it actually helps us to understand that what are the most prominent mutations, the mutations which are very likely to happen. So essentially, if you think about the landscape, what are these initial set of mutations which speeds up the process of evolution, as opposed to those mutations which are late, but at the same time can have a higher fitness. So we have more of these time resolution that way, like exactly which mutations emerge or appear relatively earlier versus those which are appearing relatively late, and which mutations can generate more mutations and which can make a species more fit. So essentially, all these informations are that way because of the fact that we have the more mutations can be utilized, and that can actually, in principle, inform us about the dynamics. Now, as one thing to say, of course, this was a teamwork, and so Yanmin and Joao, so it was like they were initially, so they were a part of my group that way, and it was a combination of computational chemistry, evolutionary biology work, so we needed expertise from both the sides. And one more important thing to mention, like when we think of evolution at the level of abstraction, if we think of, so evolution can be thought of at the level of cells, at the level of organism. Here, what we are actually concerned is more about the molecular evolution. So essentially, these are the events. If you think about the structures, if you think about the dynamics, these are more like sub-second. It's like microsecond, nanosecond, to sort of probably in minutes, because it's also about these metabolic regulation. So it's pretty fast, a kind of a dynamics happening, and we are primarily here. Our exploration is essentially at this level, where we are primarily focused on molecular evolution, but in principle, it is more about a way in which we can connect the genotype-phenotype relation. Now, again, that is another reason as to why bacterial systems are very interesting, because bacterial systems allows us to essentially have, because it is like one cell is one organism. So in principle, we can actually deploy the strategies to essentially have a complete genotype-phenotype relation. So this was actually our strategy. So on one hand, we wanted to essentially come up with a drug, and these were essentially the mutations on which we focused. So why? Because these were the mutations which emerge most prominently. These are the most rapidly appearing mutations, and these are the ones which essentially contributes a lot to the fitness. Essentially, in a condition when the cells are growing when the cells are stressed with the drug. So we wanted to come up with a compound, a chemical solution, which would be interacting with not just the wild type, but also can potentially interact with these kinds of mutations, or these kinds of mutated species of the wild type DHFR. So it was in that way more of a multi-targeting by design, but multi-targeting in a way like where we knew that how a protein can kind of mutate and incorporate that information in our design protocol, and sort of essentially be prepared for the most probable escape variation and have an anti-escape strategy. Essentially, if the drug can interact with these mutants, so essentially at least this mutation would not be the escape solution. So that was what our objective was. The second was also to understand that, okay, these are the very fancy things which we are doing at the cell level and maybe at the compound level, but within the cell, is it so that the molecule is just interacting with DHFR, or the phenotype which we see, which could be essentially more about resisting the blocking the emergence of this escape or this escape phenotype? Is it just associated with our target protein, or it's essentially a combination of a target protein with some other protein? So which is where we wanted to have a thorough understanding of this intracellular mechanism of action. So essentially, as I mentioned, this was the work got started in this way, and this was more of a spin out, which was what we were doing with our collaborators. But this is more about the framework, and we screened a large library of compounds, which was close to 1.5 million compounds, and then we did different kinds of docking simulation, allowing the protein to undergo different kinds of conformational flexibilities, because that's what happens in reality. And then also taking into, and this was like pre-AlphaFold time, so it was primarily either doing modeling or essentially taking the structure from the PDB, where we have the DHFR. We have essentially the coordinates, the X-ray, mostly from the X-ray structures for DHFR and the different kind of complexes. And this is kind of a very important phase, because it actually allows us to understand how the molecule fluctuates, because that is extremely important. Otherwise, because when we think of a protein, it's more of an ensemble of conformation. If we just design a molecule against one static structure in mind, in reality, the compound just may not bind, because we are taking a representative from these entire ensemble. So it was important to understand the distribution and essentially the most commonly sampled conformation, and generate the active site based on that, and then redock, do all these simulations one more time, and this step was computationally more expensive because we were doing molecular dynamic simulations. And then finally we had a short list of compounds, which we finally went on to do the biological experiments, biological testing, and we had this 15 and 17 series, which actually emerged pretty good. I'm not going to go into the technical details of this. This is the experiment, so which is where we do a fluorescence assay. So essentially, when we see, and it's like whether it's essentially to compare whether the way it works, if the compound is inhibiting the protein, then there would be a drop in the fluorescence, and if the compound is not, then there would be, we would see the rise in the fluorescence. So essentially, it's more with respect to the fluorescence readout. And our interest was to essentially have a protein which can interact with wild type as well as the most common escape variants. And that's what we have compared here. And finally, what we found, like this compound 15-3, it is one of the derivative of compound, which was capable of interacting with the wild type as well as the most most commonly appearing escape variants. So we went on to do. So this is something which was where we were actually using. So this was all from large library screening, doing conformational sampling, allowing the molecule to interact, doing docking simulation, doing biological experiments. But now the most interesting thing, and what happens, so this is all evolution informed, but and it's kind of, okay, fine, we know that these are the most commonly appearing mutation. In reality, now if we use this compound, which is kind of showing all good things, at least with interaction. But if we now allow the compound and allow the cells to grow in the presence of this compound and see and essentially monitor what is happening for a period of close to a month, a little more than a month, what is actually happening. Now, this is what we do in our lab. So essentially, we do real-time evolution experiments. So essentially, this is kind of a robotic setup where we actually allow the cells to grow. And this is something where we actually do things in multiple replicates. Essentially, we have multiple trajectories. So we allow the cells to grow in the presence of a drug. We also run controls, so which is, of course, trimethoprim, against which, all the mutation data was primarily derived from that. And what we observed, like when we performed these long-time evolution experiment, we found like we did not observe, that was very interesting, any point mutation. Now, the FOLA is essentially the gene which encodes this protein DHFR. So what we observe is red tress is essentially the median tress for our compound. So what we observed, like there was, and this is essentially the rise in the IC50, which is essentially the measure, like to what extent now to what extent the cell is now resisting the drug action. So there was a little uptick, which means like now the starting concentration of the drug we needed it eventually got higher. So cells adapted, which was why we needed more concentration of the drug. But the uptick essentially we observed a stagnation. So we had an explanation for this, which I will show. And what we observed that this was primarily happening, this was not a point mutation. Now, that was very encouraging. On the other hand, so essentially we could block the DHFR mutation. On the other hand, we have this trimethoprim, which is more of these traditional formulation. We observed that there are these kinds of point mutations happening. So on one hand, we were very successful in a sense that we could block the point mutation, and our robotic experiment eventually worked out. But then the little resistance which we see, so we went on to do the whole genome sequencing. Now, this is what is kind of getting even more accessible because of what we call as the genome scale science. So it's not just about the genes, but genes constituting the bacterial genome, but we also know their positions, we also know their dosage. So we have a thorough understanding of these bacterial genomes. So we went on to do the whole genome sequencing, and we found that a region of the genome essentially has higher dosage. Now, the depth height increment means like we have more gene. So essentially, the expression coming from this gene essentially is higher. It's essentially kind of twice. So the gene dosage is essentially higher. So and this is primarily what we observed is because of its dosage is higher because that segment of the genome got duplicated. Now, when we went on to analyze, so this is what we observed in this E1 and E2. These are those evolved phenotypes. When we went on to zoom, and when we further zoomed in and analyzed the constituent genes, and what we found that actually this duplicated segment is primarily constituted by transporters and pumps. So whatever the little resistance we observed here is actually happening by a very generic machinery, which is like the drug is getting pumped out. So it is not the most optimal solution, because these kinds of things like pumping out the drug is kind of non-specific. It's not just pumping out the drugs on a selective basis, it's also pumping a lot of other things. So which is why the IC50 could not go beyond the point. So it is relatively more fit. It is an adapted, it is an evolved population compared to the wild type cell, but it's not that evolved as we had otherwise seen, as we have otherwise seen for these kinds of trimethoprim resistant strains. So of course, and that is the reason as to why now compared to the wild type, the evolved strain only requires 2 times higher concentration of the drug, as opposed to the trimethoprim resistant strain where it needs like close to 100 times more. So it's like orders of magnitude higher in terms of the concentration. So we were successful in terms of blocking essentially constraining the evolvability, blocking the point mutation. And we also had an explanation that what explains this resistant phenotype. There were surprises. All this thing was good. This was essentially when we were communicating our paper in eLife. But when we went on to essentially analyze what is functioning inside the cell, we found this is the strategy. So essentially, if you overexpress, now if you have more functional copies of your target protein, ideally that would completely rescue the growth inhibition. So essentially, this would have been like this, not like this. But what we observed that the growth rate is still kind of low at a relatively higher concentration of the drug, but it's not that bad as our black dress. So which means overexpression of our target protein. Now we have more functional copies. So which means there are now unbound protein fractions. So those unbound protein fractions can actually function and can do their proper function. But that's not sufficient for the cell to have a complete rescued phenotype. So this is actually possible. And this is another experiment where we actually showed that we needed a functional protein, not a defunct protein, so for this rescue. But what we could actually infer that it is primarily happening because of probable second target, off-targeting thing. So essentially the compound is now targeting not just one protein, probably 2 protein, which is why overexpression of one target protein is not leading to the full rescue. That completely makes sense, but we didn't know where the other protein is. And this is where we actually, again, took the genome scale science approach and more of a zoomed out systems level approach with large scale data. So we went on to generate omics data. So essentially, we went on to compare drug-treated and drug-untreated cells, and we went on to essentially look for the metabolites. So because this compound, so as we know that DHFR is, as we have seen, it is a part of folate metabolism, it is a critical protein, so it would create, any inhibitor for this compound would actually have a metabolic disruption. Now, if we compare the disrupted with the undisrupted set, and if we can and kind of see the essentially compare the two from the comparative metabolome, we can actually come up with those subnetworks, essentially those metabolic pathways, which are kind of differentially perturbed, or which are kind of, in terms of their abundances, which are different upon treatment when we compare it with the wild type. So that is kind of extremely helpful, essentially to take a very zoomed out way, completely unbiased thing. Then we did this very interesting thing, which was what this is again something which we worked on for a period of around a year or so. So we kind of pulled all the available antibiotic resistance, all these kinds of metabolomics data, this metabolite abundance data, and here we applied this logistic regression model. It was more about a multi-class kind of a classification. So we had our compound data set essentially upon perturbation, and we went on to compare with other perturbation data sets. So why this is important? Because we wanted to assign identity. So let's say we have a drug which inhibits DNA metabolism. We have a drug which inhibits cell wall biosynthesis. We have a drug which inhibits, let's say, something with protein translation. So there are these certain families of drugs. So we took representative members of each of these families of drugs and went on to compare essentially the perturbation type with our compound. And this was a way to assign identity. And we found that our compound, again, by nature, has a very antifolate signature. So why this is important? Because otherwise, if you think about just the omics data, there will be perturbations here and there. Because we have to think in terms of the network. The effect within a protein doesn't just remain confined in its immediate proximity. So there are other pathways which are also going to get impacted. So it could be more of a percolated kind of an effect. So just doing metabolomics, just looking at the omics data, it's still qualitative. So this essentially this machine learning based screening in principle helped us to narrow down and assign identity, but the perturbation is very folate-like. So we focused on to folate, and then we did this metabolite. This is a very interesting mix of experimental and computational modeling, which we do with the Paulson group at UCSD. So essentially, our model informs that essentially these are the reactions, these are the subnetworks which are likely to contribute to the perturbation. So we externally supplement, we externally use those metabolites. We do experiments and see to what extent the model's prediction is correct, and it's a way in which we can actually add robustness to the model. It's more about a validation, and that's the way in which we further went on to essentially narrow down our search process. So what otherwise started off with 886 metabolites with multiple pathways eventually helped us to confine to like one pathway, which is essentially folate pathway, and in principle, essentially to just scan those proteins. So this is what exactly our metabolite abundance data is, and we can see that this is where we have our compound. So compound clusters very close to the trimethoprim induced perturbation, which completely makes sense. It has also these are like different time points. It also has overlaps with other antibiotics. That's fine, because as I mentioned, like the perturbation would also have some generic signatures, and which is where many different kinds of perturbations will have overlapping kind of trends. So as it helped us to narrow down to one particular pathway, so this is what we did very interesting, and this is what we are also generalizing. So we knew the structure because the compounds were designed against DHFR, which is a folate pathway protein. We went on to look for proteins inside this pathway, narrowed down, but now we are focusing on folate pathway, which is structurally similar, because there could be off-targeting. A compound can get confused if it actually has two similar looking proteins in the proximity. And if we use much higher concentration of the compound, so it can, in that way, under that kind of a situation, it can essentially interact with both the proteins. And that's what was actually happening. So this was this particular protein, this is a kinase protein. It's located slightly upstream to DHFR. This is where we have DHFR, and this is where we have HPPK2. And it is actually at a point where the two sort of reactions are kind of converging. So the molecule, essentially, these two proteins are pretty much enclosed. And now, in another work, we have actually shown that these proteins essentially form a large supramolecular cluster. So essentially, when the compound is interacting, it's actually, these two proteins are pretty close. So it actually can interact with both the proteins because the active sites are kind of very close. This has an evolutionary meaning because the two proteins had kind of had kind of diverged from a common ancestor, which I'm not discussing. But essentially, this strategy helped us to sort of narrow down on a specific protein. And we also went on to overexpress other proteins. So overexpression is again the strategy where we have more functional copies. But now, instead of those 886 metabolites, like thousands of proteins, we're just working within, let's say, 14, 15 proteins. And we found like the overexpression of these proteins, with which the DHFR has structural similarity, leads to full rescue. And we performed, this was again like a genome scale modeling, and this kind of shows which had partial rescue, which had full rescue, and which had no rescue. Certain things were predicted from the model. When we did experiments, we found like it's not actually overlapping with the model, but actually it helped add the robustness to the model. And also another hallmark, and this is a very convincing thing, it comes from confocal imaging. It shows like, so when this folate pathway is kind of perturbed, one of the hallmarks of these kinds of folate pathway perturbation is this, like it's actually one of the hallmarks of antifolate stress. That cells essentially starts to have morphometric changes, and what we observe when the cell, now when we actually overexpress the target protein, we actually see that we don't see that morphometric enlargement. So also from a confocal microscopic perspective, we actually found like we could actually locate we could locate and we could kind of validate the target. And this is also some enzymatic assay that's more about a kinase assay, which shows like, because if the protein is kind of inhibited, the kinase activity is going to go down. And as a result, essentially, there will be free ATP, and the free ATP would essentially lead to chemiluminescent signal. So this is what we are finding, and so this kind of is kind of the summary of where we are. So we essentially wanted to have a compound which would be broadly anti-escape, and that worked. Our evolution strategy also worked. But since it was by design kind of broad, it had these, it was also having off-targeting issues, and we generated this framework, and this framework is now more of a generic framework which can be used to understand molecular targets. So I will not, I'll stop here. These are some of these, this is something which I discussed. One thing I would like to say is now we are taking this strategy for in a very different context. So instead of metabolomics, we are using phosphoprotein, more of a phosphorylation or phosphoproteomics kind of a data set, and in the context of neurodegenerative disease to also locate the possible off targets. And this is actually in the context of Alzheimer. This is a work which we are doing at LSP. We're actually deploying this framework for off target identification. And these are the ways where we have actually applied these kinds of systems and evolutions angle. And we have used it in the context of the spike protein, the corona spike protein. This was actually a work which we performed with. This was like a joint thing which we did with as a part of CSIR's national initiative. And we are also doing sometimes this kinds of systems evolution, biological physics interface kind of an approach in very different proteins, like proteins linked with cataract formation. And this was like the work which we published in eLife and Nature like last year. So yeah, so on one hand we have evolution informed strategies. On the other, I'm not going to go discuss all these things. On one hand we are discussing this. We have these evolution informed strategies. We have now a better way to understand, essentially model this different kind of contributing factors which can actually which can help us describe the resistance. So it's more about resistance modeling. And of course, our interest essentially lies at the interface of folding and evolution. And this is what we are, as I mentioned, this framework which we have developed has a more generic use that can be extended to drugs in general. And we're also using these kinds of integrated strategies for disease modeling, and which is actually something which we do with Zion Therapeutics. It's more in the field of translation. There are certain proprietary issues, which is why I cannot discuss the research which we are doing, but also where we are in principle taking the same strategy. So these are essentially, this is going to be also the kind of work which which I would be doing in my group, and also I will be an affiliate with Harvard, so essentially it would be more of an integrated thing which we would do at Pilani-Hyderabad and LSP at Harvard with our collaborators. So thanks also to the group this has been more of a teamwork, and these are like extremely special members, extremely special people in all the science which we discussed. So yeah, so I will stop here because anyway I'm kind of beyond the time allocated, but thank you for your patience.
Thank you. Thank you so much, Dr. Chowdhury, for the wonderful presentation. I hope, the audiences, they have learned something of value from your presentation, and I'm very much intrigued at the application of the machine learning, because that is where we mostly from the CS background we work on, and to see that how ML is actually being implemented in real life, specifically in therapeutics and drug discovery, and how we can challenge the diseases and all. And it's like it is a validation of our own, like, of the research that we do basically. So I would like to congratulate you from the CS community, on behalf of the CS community, that you were actually applying these principles in order to enhance the drug discovery, or like for the benefit of the overall biological thing.
Another interesting thing I will say, like, we also, now this is more of an ongoing thing. So, like, let's say, like, this was actually a problem during COVID. I mean, let's say you have certain disease for which you don't have sufficient information. Like, let's say you know a protein which is associated with virulence, or a protein associated with, let's say, a particularly escape variant of cancer, but you don't have sufficient mutation information, which is something we need for this kinds of prediction. And if we can take some generative approach to essentially understand the kind of mutational possibilities, and you can actually think of, you can be prepared with, this is something which we are doing in the context of immunotherapy. You can come up with a cocktail of compounds and like antibodies which are primed, which are kind of pre-primed. So they already know what kind of, and as if we are shaping the evolution landscape. So as if we are putting some kind of a stress, and we know what would be more of its escape strategy, and we already have that model. So the next therapeutic thing, it could be antibody, it could be drug, in principle can actually interact with these escape variants. So as if it's like not artificial, it's more like redirecting evolution. So that is something which we are also doing. And these are really fascinating use of, like, where you do this complexity theory modeling, and like you come up with machine learning based approaches. So yeah, then we have a lot of these exciting things going on. Yeah.
Nice. So we would like to open for a very quick and short Q&A, because we have exceeded the time limit. I believe one question already came. Is there any relationship between antibiotics and cancer cells? Please give your insight. So can you please, like, in a very short way, just
yeah. So antibiotics, so essentially when the cancer cells essentially become drug tolerant or drug resistant, it's exactly the same evolution mechanism. So essentially when we have a compound, so that the anti-cancer drug is actually targeting a particular protein, which is a kind of an oncoprotein, which is kind of selectively present in the cancer cell. So essentially the cancer cell will try to escape that drug action. And when it escapes, then essentially that chemical compound would not function. So that's the reason as to why we think of different kind of cocktails. So precisely, but the only difference between cancer evolution and the antibacterial and anti-cancer drug evolution is this, that antibacterial, which is why antibacterial things are a little more simpler. Because in the context of cancer, we have this in the system of cancer, we have this concept of driver and passenger mutation. So whatever the mutations which we were discussing, they are kind of, in bacteria, we don't have driver passenger thing. So it's like one mutation driving the entire event. In cancer, what happened, like this hop from one peak to the other, it's not like to the adjacent peak. The hop can happen from, like, let's say, peak 1 to peak 6. It's because when the driver mutation is kind of resulting in a certain phenotype, there are certain associated mutation, which suddenly changes the overall phenotypic fitness to a large extent. So this concept of driver and passenger mutation makes the evolutionary landscape in the context of cancer a little more complicated. So that's the reason as to why we want to understand things in the context of bacterial system and then take it to more complex systems, and which is also a part of my own plan that way.
Thank you, Dr. Chowdhury. Anyone else has any sort of question? Maybe a very quick session. Okay, Swaminathan has raised a hand. Swaminathan, do you wish to ask something?
Yeah. Yes. Thanks for the talk, Dr. Sourav. So I have I don't have a biological background, but I try to follow whatever you said and whatever that was there in the talk. And I have a small question. So from what you have told, I inferred that at this point your research work sort of like the alteration based resistance is sort of becoming null right now with your research. But do you think that even the transporters and pumps, they could also be solved in the same, pathway, evolutionary pathway, or is there, a combination of whatever your research work has done and that combined could lead to a better result?
Yeah. It's a good question. So, in general, it was a way to constrain the ability of a cell to evolve, which we say like constraining evolvability. Now, as you mentioned, like for like each of the proteins, so essentially the complexity increases is based on how a protein essentially participates, because everything is happening in network. For metabolic proteins, the networks are sort of well defined, and we know that way, like if you have a perturbation in one of the subnetwork, how it would actually have a cascading effect on the overall network. The ones with the transporters, which we observed, like with the evolved strain. So that kind of mutation is that kind of a solution is a more general solution, and it's not an optimal solution. There are ways, and that's what we are actually trying. Why it is not the most optimal solution? Because if it had been optimal, then essentially the IC50, which we use as a kind of a number to see to what extent now the cells are kind of resistant. So which means like if you have a very resistant cell, you would need very higher concentration of drug, as we have seen for trimethoprim. That was not the case with this evolved strain where there were only this duplication and transporters were helping. It was only by a factor of 2. It's suboptimal because these kinds of transporters transport out not just our compound, but many things in general. So it works out in the favor, in a sense, like cells can still grow because drugs are getting pumped out, but it is also having other kind of, associated toxic effects, because it is also pumping out some essential things. So that way it is not the most optimal. What we can do, and what we are actually planning to do, there are some generic pump blockers which are used in different contexts that way. And since we have the genome already scanned and we know what are the genes which are there in this duplicated segment, we know what are those transporters. We know exactly the identities of those transporters. So we can actually think of using a combination. So essentially, just like the way we are using this compound 15-3, we can use that anti-transporter, generic anti-transporter, and see what happens. Probably there would still be some other escape strategy. Maybe it would be even less optimal. So I think it's that way it is completely possible, and we are working in that direction. So I think that's a good thing. That's a good direction. Good question.
Thank you, Swaminathan, for the question.
Thank you.
Anyone else has any question? Okay, if not, then maybe I'll ask a very quick sort of question to Doctor. It's not a question; it's basically a silly, a childish kind of thing I want to ask. Because I have been very intrigued with this particular thing. It is not with respect to your specific work, but sort of evolutionary. So you talked about molecular evolution, like evolution at the molecular level. I am very intrigued with this idea that how does evolution works at the molecular level. Now that's a very big topic. My question is is it suppose I'll give an example, it will be better to understand. Suppose today I am thinking that I wish to fly. What if I had wings? Okay. Now I'm thinking it very hard. Of course I do not have wings and I cannot fly. But I am thinking so hard and throughout my life, or I have thought it. Do you feel that to my offsprings, whatever the gene I will pass, there will be some molecular level kind of change will happen, so that ultimately maybe some wing-like protrusions will come or something? Maybe not the immediate offspring. Does thought give way to evolution at a molecular level? It's a very sort of abstract thing I ask, and maybe a childish also. But
it's a little philosophical thing. The thoughts, of course, do not percolate. If you think in terms of inheritance, but generally, if you think of again going back to the textbooks and going back to the philosophical perspective, that when we learn certain things, that's not exactly a vertical inheritance. So that's more of a Lamarckian kind of a thing. So if you're thinking that way, and or maybe let's say it's more at the level of execution, and so it's not something genetic that way. It's more about the things which are that way visible. But that's a way in which this kind of Lamarckian kind of inheritance. It's more like it's non-vertical that way. More of a horizontal kind of inheritance can happen. People around you can actually pick it up, but that's not going to go through it's not going to come through vertical. It's not a vertical way of.
I understand. Anyway,
there could be a long discussion on this. And
of course, it's sort of falls under it. So, anyway, so I believe that we are at the end. Any more questions do we have? No, right? Okay, so Dr. Chowdhury, thank you very much. I will also, on behalf of all the audiences present and on behalf of CSSBC, IEEE Kharagpur section CSSBC, I would like to thank you heartily for taking out your valuable time and presenting this wonderful talk. We have a short token of appreciation that we would like to virtually present to you. So if you can see the screen. So this is a short token of appreciation that we would like to present to you. Thank you so much, Dr. Sourav Chowdhury, for delivering this talk on this day, 28 February 2024. So this thing will be sent to you via email, like maybe in the next 2 to 3 days I will be sending to you. But this is, we are formally presenting the memento to you.
Thank you so much. Yeah. And I will be happy to, like, if any one of you is kind of interested in like more extended discussions, and also I'm starting my own group. So if any one of you is interested to co-ideate, I'm always happy. So I'm looking for like new approaches. So yeah.
Yeah. So I will be, if you do not mind, I may be sharing your mail ID to interested participants, so that they can directly get in touch with you.
Yeah.
So all those who are interested may directly contact IEEE CSSBC. You can just mail us, and we will give you the contact details of Dr. Chowdhury. And finally, in the final, as the final part of our talk, we would like to take a group screenshot. Okay. So all those who are present here, if it is possible, please switch on your cameras, and we will just take a screenshot. Yeah. So I'd like to request Kumarjit. So Kumarjit is our secretary, by the way, Dr. Chowdhury. He is present.
Oh, yeah.
So I'd like to request Kumarjit to take a appropriate screenshot, maybe 2 or 3 screenshots, and you will let us know once you're done.
Yeah, thank you.